I recently work as a Marie Curie postdoctoral researcher in Aberystwyth University. So it's in UK. So most of you might not know where it is. So when you look at the map of United Kingdom, we come from, our university is located in the West Wales. So it's a coastal town. It's Aberystwyth. So this is the university. So within the university, we have a very large department, IPERS. This is the department where I currently do my research. It's Institute of Biological, Environmental and Rural Sciences. So we have a very large department within the university, and currently the main goal of our department is to focus our research in response to certain global challenges which are associated with the food security, bioenergy and climate change. Since I will be discussing about the metabolomics and gene editing techniques in this webinar, I would like to give an introduction about what all facilities we have in our university. So we have two great facilities which are very much essential for doing plant research. That's National Plant Phenomics Center, which is NPCC, and a high throughput omics platform. I will first share about what NPCC, what we are doing in NPCC. So being a plant biologist, one of the most important or a difficult task is how to determine the behavior of the plants. So plants, they respond to the environmental conditions in different way. So it's very difficult to monitor the behavior of the plants, like how a plant responds to salinity stress, how the plant responds to drought stress. It not only in that situation, there are situations where you need to understand the whole development of the seed, starting from the early seed germination to senescence. So to monitor the behavior of the plant is what you call phenotyping. So being a plant scientist, it's very difficult task. So what we offer in National Plant Phenomics Center is that we have a whole platform, very huge advanced platform for doing non-destructive imaging of plant, where you can study the behavior of the plant under normal condition, under stress conditions. You can study the whole life cycle of the plant. So we have a range of small to large platform. And here I would like to show you a video, a small video where you can see how the plants are being imaged within the facility. So now I will pause this presentation for a minute, and I'm switching to a video. So kindly bear with me for a few minutes. I'm just trying to share the video now. So can you share? Can you see the Phenomics video now?
Yes.
Okay. So this is how it works here. So when you grow the plants, you can grow in large. You can grow in single pot. So each plant will be monitored for the behavior, or you can image the plant. So through this platform, it makes the life easy for the plant researchers. So you can see here, it's automatically being moved. So you don't need any manual labor. It's all been programmed. So it is automatically moving, and then it goes into a platform where it is being imaged. So that's how you can screen. So when you're working with a large population, for example, you are screening a large population of rice varieties, and you're trying to find the elite cultivars or varieties which are resistant or which are more tolerance to drought or something. This kind of phenomics platform makes our life more easier. So I just want you to show how things work here. So I'm switching back to the presentation. Yeah, so here we are. Right, so now I'm back to my slides. Is all okay so far? Okay. So, yeah. So now you saw that we have all the facilities where you can monitor the phenomics or phenotype of the plant. So from this image, it is possible to measure so many things like the leaf area, height, the chlorophyll. Even if you are working with the drought experiment, it's possible to measure the water use efficiency, the relative humidity. So there are so many things you can get from this data. Apart from that, you can also do the root phenotype, and we have a gravimetrics platform where you can understand how the plant behave in limited water conditions. So this is one of the greatest facilities we have here within our university. So next, apart from the Phenomics platform, we have an advanced imaging unit. So now when you're working with the crops like rice, wheat or maize or oats, and if you want to understand the quality or the seed traits, like you want to understand the seed color, seed shape or seed anatomy in depth, we have an advanced imaging facility where we use micro-CT imaging analysis. So this is some of the three-dimensional image which we captured here in our facility. So if you want to understand the panicle structure, the seed structure, we can do everything in here. So the second facility we have here is the high-resolution metabolomics platform. So here we have a great well-fledged lab where we have a wide range of high throughput mass spec, LCMS instruments in order to do the metabolomics, where you can identify the whole metabolome within the organism, the proteomics, lipidomics. So all these facilities, like combining the phenomics and the metabolomics, is one of the first or the basic step in determining the gene editing strategies for the future crop improvement programs. So as I said, this is an era of omics. So you have so many omics going in around in the plant, or whether it's in the animal or in other systems. So you have the phenomics. It goes to the metabolomics. So what is phenomics? So phenomics, like I said, it's how the plant or an organism behave or how it looks to a certain environment or under normal conditions. So that is phenomics. So phenomics or phenotyping, where you can monitor the changes through certain imaging techniques, through sensor methods, like how we do in our phenomics facility. And after you monitor the phenomics, then you look into the genomics, where you look in the genomic DNA, like through genomic DNA sequencing, genome mapping. Then you go again into details where you understand the function of the gene, like which gene is giving that particular trait or particular function to the plant. Then you come to the more again into the products like proteomics and metabolomics. So metabolomics is the final stage where you understand the expression of the low molecules which are being produced in response to the certain gene function or certain in response to the certain regulatory pathways. So if you scan this metabolome of an organism, it is possible for you to connect to the phenomic trait of the plant or any organism. So these are all connected. So whatever you are seeing down here will be reflected in the phenome of the plant. So in metabolomics, you use a wide range of analytical techniques, which I will give you a brief introduction in the further slides. So how you can get more insights into the plant behavior using the metabolomics, so that you can build a gene editing pipeline for further future crop improvement. So here you can see, as I mentioned before, the plants or any organism, they respond to the environment, and that changes is reflected in the genotype. For example, sometimes a plant has to survive in very harsh weather, like high, very heat stress. They has to tolerate very low temperatures. Sometimes they has to they have to survive in very droughted conditions or in more waterlogged conditions. So these are all behavior of the plant. A plant is in turn reflected in the genotype, which in turn is reflected in the phenotype. So these are always connected. They are all related in a way. So how you can connect the metabolome to phenotype? The first study is you have to understand the metabolome of the plant. So metabolite is an integral part of plant development and adaptation. So when you say metabolite, the plant itself has primary metabolites and secondary metabolites. So the primary metabolites are more used for the growth and development of the plants, like they are used for photosynthesis, respiration, whereas the secondary metabolites are more like alkaloids, flavonoids, or phenolics, which help the plant to survive in response to various stress, like abiotic or biotic stresses. So how we can do the metabolomics in a plant system? So before I explain about the metabolomics pipeline, I would give a small introduction what is metabolomics exactly. So it is a large-scale study of metabolites which is present in the cell or a tissue or within an organism. And what is a metabolome? So metabolome is a total metabolite that is present within the biological sample in response to the normal or stress conditions. So as I said before, there are like primary metabolites which are necessary for the growth and development of the plant, and that include carbohydrates, amino acids, and lipids. And secondary metabolites are those which help the plant to survive the stress, and these are like flavonoids, alkaloids, and terpenoids. So by studying the metabolome of the plant, it gives us an understanding that how the plant behaves and what is the physiology and how the biochemistry of the plant is modulated under the stress conditions. So I'll now give you a brief summary. I will try to be as simple as possible because it's a very complicated, if you go into details, it makes it so complicated. So I just don't want to make it so complicated. I want to give you the idea how it isn't easy, but to give you an idea how the metabolomics pipeline is working within the plant system. So let's say we have an experiment. We have an experiment where you have a control group of plants, and now you want to check certain plants under certain abiotic stress conditions. So you have like two groups of plants, like control plants and abiotic stress conditions. So now you want to extract the samples from these two groups. So this can be like whatever tissue you want to use. You want to study the metabolomes. It can be leaves, stem, root, whatever. And then you extract the samples from these plants, and this will be extracted in a chloroform methanol water solvent system. And this extract will be used for analytical process where the metabolome will be scanned using various analytical procedures like GC, gas chromatography, HPLC, LCMS, and NMR. So when you do metabolomics, there are two approaches you can do. One is targeted and the other one is untargeted. So targeted means, like in this extract, if you are looking for certain class of compounds, like if you want to study only flavonoids, if you want to study only the level of amino acids, that can be done through a targeted approach where your metabolome will be scanned especially for those kind of compounds against the standards. And second is untargeted, where the whole metabolome will be scanned. So you will get around like 20,000 metabolites. So it will be scanning like a 1 microliter of your extract. And from that 1 microliter, you will be getting around like 20,000 metabolites in a complete scan. So from that, you can then look for your different compounds, and you will get an idea of all the pathways. So after it runs through the mass spectrometric analysis or the analytical part, then comes the data analysis. So you will get certain, you will get the data from here. There are certain online software which you do the data analysis. And from this data analysis, you will visualize the data through PCAs and heat maps. So so these are all the preparation part. This is the sample preparation part. This is where the metabolome will be scanned in the analytical instrument, like mass spectrometry based instruments, and then data will be analyzed through online tools. And then this is the visualization part where you see your data in terms of PCAs or heat maps. So once you once you get your data, you will first do a PCA, which is a principal component analysis, where you try to understand, because first you need to know how the control and the experiment group is looking, whether it is similar, whether it is different. So this will give you an initial impression of how the control group is different from the experiment group. So let's say the blue one are the control group, the plants from the control group. So the plants from the control group, they are clustered together and they are formed as a single group in this PCA. And second, the experimental group are clustered together differently and they are distinct from the control group. So this gives an idea how different the control and experiment group are. After that, you will select the major source of variation. So what are the difference? What are the metabolites which are causing difference between these two groups? You will look for these compounds and then you will annotate these compounds using a database or the library, metabolomic library, which is available. And then you identify the compounds. You identify if it's sugar, if it's amino acids, if it's any secondary metabolites, and then you identify the compounds. And after you identify the compounds, you will visualize it as a heat map. So heat map again gives you how much they are expressed within the control group and the experimental group. So here, if you can see, this is a heat map where you can see how the control group is clustered together. So there are certain metabolites. These are some metabolites which are identified. And you can see in the control group, these metabolites are highly or they are more accumulated than compared to the experiment. So this is how you visualize the data. And once you know these compounds, then you can correlate this to the transcriptome or genome information, and you can find out which regulatory pathways are associated, or what are the pathways or what are the genes which are giving tolerance during abiotic stress conditions. So this is the whole metabolomics pipeline. It's very simple. So you just start with the whole sample, and then finally a little bit of sample, like 1 microliter of sample. You get around 20,000 metabolites which are present within that cell or a tissue. So how you can use this platform in the plant improvement studies? So due to these climate changes, the plants are under severe threat. So they have to face so many stress in order to survive within this environment. So sometimes some plants will be happy, some plants will not be happy during the stress conditions. So metabolomics will give you an idea how they are behaving during these conditions, whether it is abiotic stress, abiotic stress, and then it will also, we can use this to elucidate the secondary metabolic pathways within the plants, within the tissues. Also, once you have the metabolomic information, now there is a promising research or an area that is currently gaining interest. That is, you are trying to correlate the metabolite to genome through a genome-wide association studies, where you can target certain agronomic and nutritional states. So this is currently gain interest. So now the plant research is more focusing, like we are trying to combining the metabolomics with the genome or the transcriptome data available of the plant. So there is a question that we are asking, why these difference in the plants? For example, I will not be responding to a stress like a same way like Vivek does. So that is the same case with the plants. So every plant will not behave in a same way to a different stress or different conditions. So there are certain variations, and there is a difference in their behavior. And what is the exact fundamental cause for these difference in their behavior? So there comes the genetic variations, and how the genetic variations are causing, it's through mutation. So in this world, we are all getting mutated, like we are mutating now because of the UV light. Our genomic DNA is changing day by day. Some mutations are like, and also the DNA will repair itself. So some mutations, they are heritable, some are not. The DNA try to repair itself. So one of the nicest example which I can show these variations are the evolution of the brassica family. The story of cauliflower, Brussels sprouts, broccoli, this all came from the wild mustard plant. So a single change in the DNA sequence of the mustard plant led to the evolution of these many varieties within the Brassica species. So how these genetic variations are possible and how these mutations are happening, how we can use this mutation as a base for our gene editing strategies. So with the advent of technologies, we were possible to sequence a human genome, and now the technology is advancing so fast that we can even sequence our genome like for $1,000. So it's that easy. So it's so advanced. But when you sequence the genome, you will get millions of reads of information. So if you see, this is a small genomic DNA information where it's a combination of ACTG bases. So when you do the genome sequencing of any organism, say like human, plant, or animal, you will get millions of reads. And within this genomic data, you will have certain regions which I am highlighted here in yellow, that will code for protein. So within this genomic DNA, you have small regions called genes, and the genes will encode proteins which will be responsible for a particular trait or function or behavior of the plant. So how these mutations happen? So it can be either deletion, so there may be some deletion, some of the bases will be deleted within the gene, or for example, there will one base is changed. For example, the C will be changing to a T. So this is all happening naturally due to some certain things, but the DNA itself will try to repair. But sometimes these mutations will lead to a certain function or a certain trait. So how you can use this in crop improvement or in plants? So the one way is the traditional breeding. So in the traditional breeding, what we do is we select the parents, like, for example, if you have a plant which is resistant to a virus, you select that plant, and then you select a plant which has high yield. So you have two trait, which is resistance to a virus and a high yielding variety, and you try to cross them to get a hybrid. So the new hybrid will have both the qualities of the parents, the virus resistant and the high yielding trait. Then you have the transgenic. So in the transgenic, what you do is you will take a gene or a gene which is responsible for a trait from a different species, and then you will try to introduce them to another species to create a new variety. So that is transgenic. That is what you call the GM crops. Another method is mutation breeding. So in mutation breeding, what you do is you will treat the seeds with the radiation, ionizing radiations or chemicals, and you induce mutations, and these mutations will itself create random mutations within the genome, and these can be related to any specific trait. But let me show an example. For example, if you want to create a seedless orange, you do mutation breeding, you treat the seeds with the mutation, and then you screen for this mutation. So there will be like 500,000 mutations happening within the genome, and you have to screen all these mutations to select this one desirable trait. So all these methods of inducing mutations or removing a DNA or altering a DNA, all these are kind of like very tedious, and it's time consuming. And how we can combine all these strategies to generate a new gene editing platform. So gene editing platform, what we do is it's more targeted. So we know where we are creating the mutation and what trait we are trying to replace. So we are trying to do a targeted way. So it can be specific edit. So if you know the genomic information of a plant, you know which gene you are going to change. You are trying to target that gene. You're making some edit and you create a new variety. So this can be either by removing, altering the DNA, or you will be adding new DNA, which is kind of genetic modification. So there is a boom recently with the gene editing technology. So one successful method which is gaining more research, more interest within the plant researchers or any researchers is CRISPR-Cas9. So that is a more efficient and simple gene editing tool. The two wonderful women, Dr. Emmanuel and Dr. Jennifer, got the Nobel Prize in 2020 in chemistry for this development of CRISPR technology as a genetic scissors. So how we can use CRISPR in plants? Before I get into details, I will try to explain the CRISPR-Cas9 in a very simple way. I just want you to give the idea of the CRISPR. So in my words, I would say CRISPR-Cas9 is searching and cutting. Like it's literally, you are searching something in the Google and you're cutting it with scissors. I'll make it simple. So you will know the genome information of your plant you're going to change. So we will look for certain 23 nucleotide sequence of that target gene. So it's like you are searching something in a Google platform. You will search something in the Google box and it will span so many, to go through so many literature or through so many databases, and it gives you the information. Like that, within the DNA, when you target these particular target gene, you search for certain 20 to 23 nucleotide sequence within the genomic DNA. So it scans a whole these millions of reads and it finds that target. And when it finds the target, the Cas9 is the scissor. The Cas9 is the scissor. So this Cas9, when you find that sequence, the Cas9 will make the cut in the DNA. So it's searching and cutting the DNA of that is cutting your targeted gene or targeted gene sequence. And then what the DNA do? The DNA itself repair. So it will give the new sequences. So this is the principle of CRISPR-Cas9, where within the whole millions of genomic data, you will search for a particular sequence which is of interest for editing. And then once you have the sequence, you will cut the DNA with the Cas9 protein. And once it's cut, the DNA will repair. So that's how you get a new, that's how you create CRISPR-Cas9. So when you discuss about the scientific terms, what are the things you need for the CRISPR-Cas9? First, you need a gene. For example, in a plant, if you're trying to increase the seed size, you need to target the gene which is responsible for that trait. So once you have the gene, then you have the Cas9 protein, which is the molecular scissors, which will be responsible for cutting the DNA. And then you have the guide RNA. So guide RNA is the 20 to 23 sequence, which will be searching like a Google search engine in your genomic database to search for that particular gene sequence. So once this Cas9 and guide RNA is in the system, is within the cell, it will target this sequence, so finds the sequence, and then the Cas9 will cut the DNA within the cell, and finally the DNA repair. So this is the whole. So you need a CRISPR-Cas9 construct which will have a guide RNA Cas9 to express, which helps in the expression of this protein within the cell. So this can be delivered in the plants through agrobacterium transformation or particle bombardment. So once you have the transformants, you screen these new varieties through antibiotic selection or any other selection criteria. So this system is currently established in Arabidopsis, rice, tobacco, and it's very simple compared to other gene editing methods. It's very efficient. So when what exactly CRISPR does? So if you're thinking what we are going to do with CRISPR. So if you remember, this is the sequence which I showed before. So there are mutations which is happening. So what we are doing is we are trying to target the mutation. So if you consider the mutation breeding, so mutation breeding, you have lots of mutation going on and you don't know which mutation is giving that trait. So it's kind of like fully random. You never know. Here, it's very peculiar. It's targeted. So once you have the cut, the DNA repairs. So it can be either by deletion. It will give a single change of the base. So if you are looking for a plant like which is less resistant to certain stress, you can edit that plant to make it more happy with the CRISPR-Cas9 technique. So it's more precise. So here you will have, which gene or which target you have, a target gene you want to edit. So you will edit that gene, and when you edit that gene, you will get this plant more resistant to particular stress or any disease. So that's where the CRISPR-Cas9 makes the breeding more easier compared to other conventional breeding and other genetic modification. So it's more simple and it's a versatile tool. It's more targeted. So how you can use all these omics method of now you have, so once you have the metabolomics, you can relate which gene or which target you want to work for the gene editing for the crop improvement. So the metabolomics helps you to find the pathways which will correlate to the gene targets, which can be used as a further step in gene editing for improving the crops. So now the major question is how you're going to use these methods in plants. What is the need for it? Why you are making all these changes in the plant? So the main question or the concern a human society is facing nowadays, how we will be sorry, I think someone's microphone is okay. Now it's okay. Sorry. So how we will be feeding the growing population. So our population is increasing. At the same time, there is a huge increase in food. Also, due to the development, there is limited availability of the land. So it has been estimated that by 2030, there will be a major reduction in the production of the major crops like rice, wheat, and maize. So we will need more crop production. But it's not about the limited land. There are so many other environmental constraints which affect the crop productivity. So we need to find a solution of how we can get a sustainable food future in order to feed this growing population. So one strategy is either you can use all these methods to improve the major domesticated crops like rice, wheat, maize, which forms a major caloric consumption for human diet. So you can either improve them, make them more tolerant to all these stresses, or increase the yield. But there is another aspect of another aspect for this, that is, we can look for orphan crops. So what are orphan crops? So orphan crops are those which are like lost or underutilized crops. So these falls under the neglected side of food security and climate change. So these are crops which hasn't got any attention from the farmers or from the research community. So there are lots of crops which have high nutritional profile, good agronomic traits, but it is not so familiar within the human within our food system, and it can be an alternative to the major crops like rice and wheat. For example, some of the orphan crops that belong to cereal groups are millets like pearl millet, bajra, foxtail millet, ragi, and some also comes under the legumes and oil seeds. Some are like root crops, some belongs to fruits and vegetables. So how you can feed the food future? So you can bridge the gap between the orphan and domesticated crops by using all these omics techniques. So now the plant researchers are so much behind to improve the domesticated crops like rice, wheat, maize, oats. But the major attention is lacking in these crops, which can be an additional supplement or an additional alternative to the domesticated crops, or that can also dominate the domesticated crop because of its nutritional and agronomic significance. So here, my research interest, what I'm currently doing in Aberystwyth University, is my focus is on teff. I don't know how familiar is this crop is, because it's an Ethiopian crop. It is an orphan crop. In India, the most similar crop to teff is ragi. So teff is an Ethiopian staple crop. This is an Ethiopian farm. In Ethiopia, they grew the teff both for their human consumption. Also, it benefits the economy. So teff itself, the word it belongs to the teff. It belongs to the grass family, and it's similar to ragi and pearl millets. So the word teff itself means lost. So that is the meaning of the teff, which means lost, because it's a lost crop. There is another also reason, which means because of its small size, there is lots of yield loss. So that means teff means lost. And Ethiopia is the center of its origin. It is now cultivated in India, Australia, and Germany. So in Ethiopia, they use the teff seeds, like how you make the rice seeds, like rice flour. They use teff seeds to make teff flour to make injera, which is a daily bread of Ethiopia. So it looks like this. This is injera. It is like a pancake. Like in our place, we could say it is similar to appam or dosha. And then teff straw is used as an animal feed for its high digestibility. So why teff? So teff, it has great nutritional benefits. So these are the teff seeds. This is teff plant. And then it has great agronomical traits, but it is a neglected crop. It is mostly confined to Ethiopia, and there is no much public awareness, or researchers are not taking much interest in order to improve this crop. So what are the nutritional benefits? So one of the major benefits of teff seeds are they are completely gluten-free. So the people who are having gluten sensitivity, alternative for wheat, and those who have diabetes can use this crop, this as a food because of low glycemic index. So compared to other major crops and other orphan crops, the teff has high levels of essential amino acids, high mineral content, it has high fiber, low glycemic index. So all these nutritional benefits makes teff as a future health food, and now it is slowly gaining interest in the European and the US food market. Apart from the nutritional traits, the other benefit of teff is the agronomic traits. So teff is more resilient to the extreme climatic conditions. They can grow and adapt to a range of altitudes from the sea level to very high altitudes. Also, they have reasonable tolerance to the drought, high waterlogging conditions, and if you have any risk if you have any risk during the cultivation of other major crops, it can be used as a catch crop in order to avoid the calamities, and then it can be used for it can be stored for a long time. Like teff seeds can be stored for a long time without any attack of pest, and it has a longer shelf life. So all these nutritional agronomic traits make it as a promising crop which can be further developed for future. So as I said, one of the challenging aspect of teff seeds are they are very small. They are the smallest grain in the world. Like if you compare the teff seeds with rice and poppy seeds, you see how small they are. They are the tiniest grain in the world, but with lots of nutritional potential. And it comes to around like 1 to hardly 1.5 millimeter in size. And when you take one wheat or one wheat grain, it equates to around 150 teff seeds. So within the teff, you have different varieties. You have white colored seed varieties, red and brown. So the white ones are the ones which are mostly used for export. It is of great economic significance. And the red and the brown ones are the ones which are rich in iron and calcium and is mostly used for human consumption, food consumption. Usually it is used in the household use. So what are the limitations in teff? So teff, as such, it means lost. One aspect is it's a lost crop of Ethiopia. And second, it's because of its smallest size. It is prone to yield loss. Apart from that, another concern is lodging. So if you compare the area of cultivation in Ethiopia compared to other major crops like maize, sorghum, wheat, teff is cultivated in a majority of the area, but when you compare it with the yield is very low compared to other crops. So there are certain limitations within the teff research because there is not much biotechnological strategies being done in teff, and there is lack of genetic resources and investment by government. It is a completely neglected crop, but it has a more significant potential as a future crop. So what my work is on Super Teff project, which I am trying to utilize the teff as a system and host to explore the genetic resources by using all these omics and gene editing strategies. So currently we are now applying all these techniques in improving the domesticated crops like rice, wheat, tomato, etc. So now I am trying to implement all these techniques in teff in order to convert this from an orphan crop to a superfood, also to increase the awareness of this healthy superfood in UK and in Europe. So the primary objective of my project is to understand the natural variation. So like I said in my initial slides, there is lots of genetic variation happening within the plants, within the animals, within the humans. And these natural variations are because of the mutations and which can in turn result in certain trait. So when you look at the teff population, you have like tall plants which are very tall, which are average, which are very short, which are high in yield, which are less in yield, which are white-seeded variety, red-seeded variety. So what is the exact cause of this variation within this teff population? So I'm trying to understand with the use of metabolomics. So once I have the metabolomic information, I will correlate this with the genome of the teff to understand the agronomic and nutritional traits. The second objective is how to apply the gene editing in teff. So one of the major problem in teff is its lodging, because teff has very long stem. If you remember, it has very long and slender stem. So when it attains its maturity, the plant tends to fall off, which is called lodging. Lodging means the stem or it will fall off. So that's lodging phenomenon, and it causes yield loss. It is the main concern in teff and is one of the primary reasons for yield loss. So we are trying to implement a Crispr platform in teff in order to address the constraints related to lodging, and we are hoping to develop lodging resistant teff varieties in future. So with all these omics and gene editing, what we are looking forward to the future is there are lots of potential within because we have around a numerous number of plants, but within the plants only 20 or 30% plants are used for human consumption. So there are majority of the crops are underdeveloped or lost or neglected, but with lots of nutritional and agronomic traits, and how you can use these techniques to improve these orphan crops. Like in my case, I have identified the teff, which is a gluten-free grain, which is highly adaptable to certain extreme environmental conditions, and it can be made as a superfood that can be an alternative to major crops like rice and wheat. So the crop improvement. The crop improvement in future has lots of potential. It's not only restricted to the major crops like rice, wheat. It can be an open to more underdeveloped crops like teff, millets, other legumes which are neglected. So that's where, and also it also helps to increase the awareness among the public and researchers so that they may become used to the new crop, because it's very difficult to introduce a new food system. Like you say, like in the market, for example, I would say the quinoa. So the quinoa, it's now, most of us all know what is quinoa and is considered as a superfood. It's a pseudo cereal, but it was not so familiar a couple of years back. But now, because of its nutritional profile, it's because it's a healthy food, people are getting used to it. So now people are more familiar to quinoa, and people are becoming more health conscious because of the changing lifestyle. So likewise, the public awareness within the public and the researchers is a very important aspect. So it's very important where you have to identify these crops And it's a very important step, how you can introduce them to a food system. So the research with all these omics and gene editing will help or provide genetic resources or information that will help the public to understand the significance or value of these kinds of crops. Yeah, with this, I would like to conclude my talk. I would like to acknowledge my supervisor, Professor Lewis, my co-supervisor, Professor Rathan, and Professor John and Fiona for giving me an opportunity to work in the Plant Phenomics Center. Before I finish, I would like to share an information to the students. So British Council has introduced a master's scholarship program for women from South Asia. So women candidates who are interested to apply for MSc or to do who are interested to do MSc in UK can apply for this scholarship. This is STEM scholarship. So this is in association with Warwick University in UK. So they have 3 courses: Masters in Environmental Bioscience, Masters in Sustainable Crop Protection, Masters in Food Security, and everything is funded. It's fully covered. You will the tuition fees, you will get monthly stipends, you will get your airfare, you will get your visa fee, and everything is covered. So whoever interested to pursue masters in UK, this is a great opportunity, which I thought it will be nice to share with you all. So if you are interested, you can either contact to the International Officer Amarjit at Warwick University. He will guide you with the application process. The last day to receive the application is next month, April 18th. And they are holding a webinar on the same, which is taking place on 22nd of March. So this is the event registration link. If anyone is interested, I could share more information with Dr. Vivek or Dr. Rashmi. So that's all. Thank you for your kind attention. I hope I made the omics and CRISPR more simple than it's not.
Yes.
Okay.
Absolutely. Thank you, Aishwarya. So it's Aishwarya had done a wonderful presentation and wonderful discussion. So I invite all participants to have a discussion on with Aishwarya. I'll do one thing. I will start with one question.
Yeah.
Actually, during the metabolomics, one of the challenge, I think, most of these traits like abiotic stress or biotic stress, most of the stress are it's multiple isoforms are involved or multiple genes are involved, and one gene itself has multiple isoforms, and lots of crosstalks also there. So. Yeah. When you check a metabolic profile before and after abiotic stress or biotic stress or something like that, whether it will be more complex or whether it is possible to get a valid conclusion about the gene regulation or something, or how the trait is controlled or what correlation. What is your...
Yes, it's a nice question, Vivek. So it's very complex. So when you're scanning for the metabolome, like I said, you are scanning for the whole metabolome. So from this, you will get around like 20 this metabolites. And the another limitation is that the metabolome for all the plants is not available. I'm speaking in terms of the plants. For example, the recent metabolome which is available in the database is Arabidopsis and rice. So that's one initial difficulty with the metabolomics. We don't have the information from everything, but when you do metabolomics, like you are doing for the stress experiments, like for example in drought, like I said, there are so many regulatory pathways involved in the stress mechanism. So one thing with the metabolome is that it will ease the task, like it will give you an initial impression, what is the significant variation between the two, like the control and the stress group. Like it will give you an initial, to give you an idea which are the metabolites that are regulated, which pathways, for example, whether it's a flavonoid pathway or whether it's the amino acids which are regulating. And from that, you can go back to the transcriptome. If the transcriptome is available for the particular plant, you can go back to that. And then another thing is, of course, you need validation. So once you find the data from metabolomics, you have to correlate it with the gene networks. And then again, you need to do a validation, which can be either through a real-time studies or some gene expression studies or some other, for example, if it's some hormone crosstalk like ABA or jasmonic acid, then you can do some feeding assay. So of course, you need some cross-validation after that. But the initial task is to identify which is which. I think metabolomics gives you an idea where to look at it.
Absolutely. And one more thing, I think, whether is there any future possibility that whether you suppose in particular condition you will get a transcriptomic profile as well as metabolic profile.
Yeah.
Whether we can compare these two things.
Yes, you can do. So there are like now single cell transcriptome, everything is so advanced now. So if you're trying to look at in a same tissue, you can go from the same tissue. For example, if you're doing the metabolic extraction from a sample, so from that, you can isolate the metabolome, you can isolate the DNA, you can isolate the aberrate from the same tissue. And with that, you can correlate everything. So that's more, it makes more easy. So that's possible.
Thank you. Students, you can ask your questions or you can type on the chat box.
I hope I explained it very simple.
Maybe no, not like
I didn't make it so much complicated.
Yeah, that's good. I think that's good because
it's very complicated. If you explain gene editing, it's very tough for the students to get through. So I think I have made clear the idea of what we can do with the gene editing and omics.
And one more thing here, the time is 2:30, 3:30. Maybe they have after lunch. Maybe they're not. Yeah.
Okay. But Vivek, if someone is interested with the master's program, it's a great opportunity.
Yeah, you type the link in that chat box, maybe straight away.
I will share that because even if I'm not sure, but if anyone is interested, just let me know. I can share the more details. It's a very good platform for, if someone is more interested in doing masters in UK, which will further help in their PhD as well. So.
Resmi?
Yes.
Yeah.
Aishwarya, I just have one doubt. You are doing CRISPR-Cas9 in teff, I think.
Yes.
Okay. So how can we target? Because is the teff sequence known?
Teff, one genomic sequence is available, but it's very limited. It doesn't has full coverage. But whatever gene which I am targeting, that has been sequenced. So I do have the information from Teff. But the initial difficulty is that it doesn't has any optimized tissue culture and a good transformation system developed in Teff. So now I am trying to optimize that. That is the challenging thing. CRISPR is easy, but the tissue culture and because if you remember in the slide, they are the tiniest seed.
Yes,
so whatever tissue culture developed is through the immature embryos. So the seed itself is 1.5 millimeter in size, and the immature embryo is hardly like. Yes, exactly. And if you want to get tissue culture from immature embryos, you have to literally dissect around like 100 embryos from the from this caryopsis, and it's a painful. So now we are trying to optimize the tissue culture with the seeds and meristem. So that's the initial thing we are doing here. And then to develop a good transformation system. There is only one article available, which is not so efficient. So before CRISPR, that is the main objective for me.
Okay, that's why I asked, because for CRISPR, you have to know the target sequence, right?
Yeah, I know the target sequence because I'm now just going with the 2 targets, which is already available, which we have some preliminary information, but that's okay. That's fine. We can make the CRISPR construct. But the thing is how you are going to regenerate and do the transformation, because there are lots of cultivars within the teff, and they are there is lots of diversity within them. So it all depends which cultivar shows the maximum regeneration efficiency, which explant, which transformation, whether it's agrobacterium, which strain. So there's lots of optimization which I have to do.
Okay. Okay, so all the best for your postdoctoral research.
Thank you.
Okay, so I think students have no doubt so far.
All right.
This is a golden opportunity. Students, please come forward if you have any doubt, or type in your chat box. These opportunities will not get you in. It is very occasionally you will get this opportunity. So please grab this opportunity and clear your doubts. Otherwise, we will end this session, I think.
Yeah. I've tried to make it simple because I know I don't want to make them too complicated, so they don't end up without asking any questions. So at least some general doubts, some simple doubts should be okay.
Okay, so I think we can.
I think Dr. Jinsi and Divya joined, I think.
Divya. Divya was there.
Yeah.
Malika is there.
Then Jinsi.
Oh.
I didn't see Jinsi. I saw Malika and I saw Divya in between. But yeah, Maju, Chereta?
Yeah, Maju.
Maju is there. Okay.
Krishna.
Okay. Nice to be here. I mean, to be back.
Okay, so one question in the chat box.
Yeah, this will go on.
Is there any unintended consequences after transformation?
There will be. It depends. So there will be. I'm not saying there won't be, because you are trying to make some changes in the plant. That can be there may be some phenotypic consequences. For example, if you do some mutations, some plants, they will become so, like for example, the growth will be retarded. So there will be some phenotypic consequences. So that's how you have to optimize when you are doing the mutations. Maybe you have to target different regions of the sequence of the gene in order to get devoid of that unintended consequences. But yes, there will be.
I think we can, Anu.
Thank you, ma'am. Thank you for the wonderful section. Now, I invite Dr. Rashmi MS, Assistant Professor at Department of Botany and Coordinator of the webinar, for delivering the vote of thanks.
Okay, good afternoon all. So we are coming to an end of the 2-day international webinar series on proteomics and metabolomics. On behalf of Department of Botany, SNGS College, Pattambi, I take this opportunity to propose vote of thanks to those who have directly or indirectly contributed to our webinar. Firstly, I may like to express our sincere thanks to our dear resource person, Dr. Aishwarya, the prestigious Marie Curie postdoctoral researcher at University of Aberystwyth, UK, for giving an excellent coverage on metabolomics and CRISPR and the methods and the techniques involved in it. She explained the advantages of CRISPR-Cas9 techniques over our conventional mutation and other breedings and genetic modifications of crops. And she also revealed some interesting facts about the high throughput facilities available in their universities, like Green Machine, for monitoring the plant growth and behavior. I am sure that the students, the budding young researchers, are enlightened with her talk, and I wish all success for her research work in teff. So I would also like to take this opportunity to place on record our hearty thanks to our principal, Dr. Saji Stephen, our vice principal, Dr. Prasanna P.K., and our IQAC coordinator, Dr. H.K. Sandosh, for the support and guidance they have extended throughout the webinar. I'd also thank all the participants, the faculties, students, research scholars from inside and outside Kerala present here for paying your attention. Thank you for attending our webinar. In order to help us continue to make our programs better, please provide us with the feedback on this webinar by filling out a quick questionnaire, and the link for the webinar and the e-certificate is provided in the chat box. Once you fill the questionnaire, it will be automatically directed the certificates to your emails. So you will get your certificate only after completion of the feedback forms. You have to fill your full name and affiliation. For students, you can type your affiliation as MSc, BSc or research scholars and mention the full address of your institutions. That will appear in your final certificates. So thank you once again for listening and listening our webinar. And thank you, Aishwarya. Thank you for your effort towards the talk. Thank you.
Thank you, Rashmi. Thank you, Dr. Rashmi, and thank you, Dr. Vivek.
So the link is provided in the chat box once again. So please go through the link and complete your feedback.
Okay. Do I need to do my feedback?
No.
Yeah. We will send you certificate.
Asking.
We will send you a certificate maybe. Yeah.
Okay. Okay. Thank you.
Okay. Bye.
We will contact you.
Once again, thank you all. Here we wind up our program. Thank you for your questions and cooperation. Thank you.
Okay.
Aishwarya, you can, because we'll continue for some time for
Okay.
getting
Okay,
for those people
okay.
feedback. Yeah. Thank you.
Okay. Thank you, Vivek. Thank you, Dr. Rashmi. Bye. Thank you. Yeah.
Bye.